2014 Albert Lasker Basic Medical Research Award for discoveries concerning the unfolded protein response, an intracellular quality control system that detects harmful misfolded proteins in the endoplasmic reticulum and signals the nucleus to carry out corrective measures. Honors Kazutoshi Mori and Peter Walter.
So I always get excited when things don't make sense, because if everything just works the way we expected, that's sort of the logical progression. But the moment you have things that don't make sense, and that repeatedly don't make sense, then it becomes exciting, because then it's clear that there's something to be discovered here, something we don't understand at all.
For decades, Walter and Mori made independent journeys to discover the core components and complex processes of an intracellular quality control mechanism.
The unfolded protein response is a quality control mechanism present in every cell in the human body, which allows for the quality control of processes taking place in that cell. And so they govern the many diverse different activities of different types of cells and are involved in many kinds of disease and health processes.
So all our cells rely on having properly folded proteins with which they can carry out their functions. And in order to fold them properly, you need machinery that needs to be there in the appropriate amounts and needs to work properly. And what we've figured out is mechanisms by which the cell adjusts the amount of this machinery to make sure that everything is just perfectly folded.
In the 1980s, insights about this machine are revealed by the research of Joseph Sambrook and Mary Jane Gething. For Dr. Mori, the timing is perfect.
1 year before I went to Dallas, Joe and Mary Jane discovered here is a signaling from ER to the nucleus. But no molecules are known or how stress signals transmit to the ER to the nucleus. Nobody knows. I sent application to Joe and Mary Jane's lab. Very fortunately, they accepted me. Then I went to Dallas and I met UPL. That changed my entire life. So since then, I've been working on UPL for 26 years.
One of the mysteries of the folding protein machine is how it can sense need and adjust accordingly.
So we started out with a very simple question: how does the gene expression program in the nucleus know what's going on in the inside of the endoplasmic reticulum? And then we worked in a genetic screen, identified components that map on that signaling pathway.
Subsequent investigations by Mori and Walter examine how IRE1 delivers the unfolded protein message.
The first component we identified and Kazu Mori identified independently turned out to be IRE1, which is a transmembrane kinase sitting in the membrane of the endoplasmic reticulum. So it has two ends. One end is in the lumen of the organelle, where it senses what's going on. Do we have enough capacity? Do we have unfolded proteins? And then it transmits that information across the membrane to eventually activate a gene expression program.
First thing discovered IRE1. It's a sensor molecule. It detects misfolding in the ER, then send the signals. Then next target is this is a transcriptional program. You need a factor in the nucleus to increase chaperones.
Again working independently, Walter and Mori set out to identify the courier that picks up the signal and carries it to the nucleus.
It worked well, and I identified the HAC1 gene.
So HAC1 is this transcription factor that activates the gene expression program according to the needs of the cell. And it is produced only when its messenger RNA has been processed by IRE1. So IRE1, when it senses in the membrane, when it senses a condition in the endoplasmic reticulum that requires activation of the program, then the messenger RNA gets cut by IRE1 in two specific spots, and then a ligase comes along and puts these ends back together. And that's a key mechanism.
These are just a few of the breakthroughs made by Mori and Walter in their lifetime of research, dedicated to unraveling the complexities of this and two other pathways of the unfolded protein response.
It's detective work. What we do in the lab is to collect clues of how things might work, and then we have to put them together into a cohesive picture of how all the different little things interact with one another, and how we come up with a cohesive model.
Mori and Walter have elucidated a process that lies at the heart of normal cell function. Many scientists are now pursuing ways to harness the unfolded protein response for medical advantages.
This sort of is really the foot pedal, the accelerator and the brake of the car in terms of many of these processes. So it will have enormous impact in dealing with human health in these diseases, many a large spectrum of diseases.
It's amazing 10 years, from 1993 to 2003. It's a dream 10 years.
Yeah, in particular, this pathway has been amazing. Every leaf we turned over pretty much had something underneath that we didn't expect. So it's a not linear road. It's a serendipitous, meandering road of discovery. It's been wonderful.